So our next speaker is a postdoctoral fellow here from the Method Cancer Center. His name is Kayode Olumoyin. I'm sorry, I'm really bad at this, but we look forward to hearing your talk on modeling adoptive cell therapy in bladder cancer using physics-informed neural networks with adaptive loss weighting.
All right. Thank you very much for the opportunity to speak at this conference. It's been wonderful so far. And thank you for the introduction to PIN in your earlier talk. So this talk, I will be using the physics-informed neural network to model adoptive cell therapy. And we'll be using data from a bladder cancer study. Okay, so the goal of my talk is that we'll use PIN to investigate how some parameters in a differential equation model, how they can be time varying due to external intervention. So in this text, we mean like the introduction of therapy. So we'll be using experimental data of a preclinical model of chemotherapy and immunotherapy. And this is the outline of my talk. Okay, so now on to the preclinical mirroring model. So this is a study that was done by our collaborators, published in 2023, where they instilled the MB49 over cell line into mouse. So in the experiment, they saw that the presence of these cancer cells caused there to be this elevated amount of myeloid-derived suppressor cells. And so they used the chemotherapy, gemcitabine, as a lymphodepletion agent. And so this is the timeline of the experiments they did on day 0. They instilled the cancer cells. They started to take measurements by day 6, and the chemotherapy was given on day 10, and immunotherapy was given on day 14. So we've included these days 17 and day 23. Those were days where histology information was collected about these mice. So this is the differential equation model we came up with. So if you remember from the talk by Anna yesterday, she gave a variant of this differential equation model. So there are 3 interacting cells. So we have the cancer cells, the MDSCs, the T cells, and then the drug gemcitabine in that compartment. So one of the assumptions in this version I'm showing is that there are no T cells in this model until it was given by day 14. And then you're just seeing the timeline of when the drug was given. On day 10, gemcitabine was given, and on day 14, the T cell was given into this model. So this is just showing you the parameters in this model. So the parameters in black, so those are the parameters we're going to try to solve for. So those are parameters we don't know from this differential equation model. The parameter in red, so that's just looking at the drug, and then from what we learned from the biologists about how long it takes for gemcitabine to clear. So we're able to compute that parameter. And then the 2 parameters in purple, those are just the injection of the drugs. Okay, so how did we initialize the model and set up? So looking at the experimental data, we can look at when the experiment measurements began to be recorded. And so looking at that data, we can compute the proportion of the cancer cells and the MDSCs at the start of the experiment. So remember I said there were no T cells in our model at the start of the experiment. And then you can see the way we computed the drug clearance. So that's just shown. And the experimental data, we use like a data augmentation method, a cubic spline approach to connect the experimental data. And so we can use that information to look at the estimated proportion of the total volume at day 17. And then we can look into the histology data and count the actual cancer cells, T cells, and MDSC cells that are present, and then use that proportion to estimate what we think is the amount of those subpopulations at those 2 time points. And that's going to be important for us. We're going to use that to constrain our model. Okay, so in the earlier talk, Sarah spoke about PIN. So I'm not going to like teach, or go into details about PIN, but I just want to say, what does PIN really do? And then how would you set up PIN? So let's say you have a differential equation of the setup that you're trying to solve. So that N could just be a differential operator. And then you have this F and G that is known at some point. So what PIN actually solves for is this other equation, whereby U sub NN is the output from PIN, but then it is solving equation 2, not equation 1. And so how you're going to accomplish that is, in Sarah's talk, she mentioned the regularization, that PIN is a way for you to set up this loss function as a regularization to make your solutions fit into some governing equations. So that's accomplished here by these two loss functions. So the residual loss, where you make PIN to satisfy the governing equation, and then if you do have constraints, as in this example, then you can also constrain the solution from PIN to satisfy those constraints. So using this as a setup, this is how we set up PIN. So the output of PIN is just the those subpopulations we're interested in, the cancer cells, the T cells, the MDSCs, and the drug. And that's really what PIN is. It's just that feed-forward network, and that's what it's going to learn. But then we constrain that using, in this case, the data loss, the residual loss, the initial condition loss, and the biology constraint loss. So I put this setup this way to show that this is different from like the vanilla PIN. If you go look into the paper from Kaniadakis, from their 2019 paper, in that case, you would have to know what a manual, to adjust the weight, because these weights are not created, these losses are not created equal. Like PIN is going to learn some things faster and it's going to do some. So we, in our implementation, we can actually adaptively learn what the right weight is for this network. And this is just showing you what each of those losses are doing. So like the biology constraint loss, I mentioned that's where we enforce those histology time points on each of those subpopulations, and then the initial condition loss to make sure PIN is satisfying what the initial proportion is for those subpopulations initially. And then just to mention here that what PIN is fitting, because the data we get, we are assuming it to be the sum of all of those interacting population. So the sum of all of those subpopulation is what PIN is matching to the experimental data. And then here is to show you that you can, in our case, since we said we want to look at some of the parameters being time varying, so it's a very, very simple way to extend PIN. So you just, you keep everything else as you have it, and then you just had like a new PIN compartment where you then learn some of those parameters to be time varying, but now in an unsupervised way. But if you do know some information about that, you could add that to your loss function. Okay, so this is the example we did. So we looked at one of the parameter, in this case, the rate at which the T cells are killing the cancer cells, KTC. So if that's time varying and every of the other parameters are constant, so what do we get from PIN? PIN is able to match the experimental data. So that's the first graph. So if you look at the sum of all of the compartments we learned from PIN and we compare with experimental data, PIN matches that. And then it's making this prediction for the subpopulation. So the first one is the cancer cells. In the middle, you have the T cells, and then you have the MDSCs. And then in addition to that, we learn the time varying parameter here. So one of the things I just wanted to see from here is that if you look at what we learned from PIN, PIN is able to learn that there is no killing before the installation of the T cell by day 14, and then subsequently following that, there is like this reduction in the kill rate, which is also matching the amount of T cells that is present, that is present in this, in this solution from PIN. Then to really understand like these dynamics, we also did like a case where all the parameters are constant. And so that's this case here. So we can see the solution from PIN. So if you look at the sum of the subcompartments also from PIN and compare with experimental data, it also matches. And then we see the sort of dynamics PIN is also able to learn in this constant case. But when we compare this with the time varying case, we begin to see some differences. We see that at the period where there is no known data, like in that period, and if we look at what PIN is estimating for the tumor cells in the time varying case versus the constant case, the time varying case seems to match when the therapy was given. And then the T cell was given, and then you can actually see that there is actually some reduction in the cancer cells, that's in the time varying case. And then if you also look at what PIN is learning for the T cells in the time varying versus the constant case, you can actually also see that there are different peaks. And then you can also see that in the constant case, there was like this extended proliferation of the cancer cells when it was given, which is not seen in the in the time varying case. And then in the MDSCs, we see that in both solutions, they tend to predict that the effect of the lymphodepletion when gemcitabine was given on day 10, you see that the amount of gem seemed to be going down in both instances, but they have different peaks that both cases are able to learn. So with all of this, we feel like, okay, we need to have more data, maybe from the point where we don't have data to actually see which of this model or approach is better. And the other thing we did was to look at different combinations of parameters being time varying. And here, this happened coincidentally. If you remember from Hannah's talk yesterday, she was showing some of these parameters that she found to be sensitive to the model in the global sensitivity analysis she did. And two of those parameters are actually in our best case scenario for the time varying case, which is highlighted in red here, which we can show in all of those parameters to have the best performance. Okay, so in conclusion, so I just want to say we've demonstrated an application of PIN where you can, in this regularization framework, you can combine PIN as you have it with different biological information you have, and you can build this sort of loss function that could help you steer PIN towards like an admissible solution. And then also shown that the prediction we observed in the experimental data, matches better when some of these model parameters are time varying. And we see that the difference in the constant parameter case and the time varying case, this informs that there needs to be more data, for us to control, to have like an approach on how to combine this biology with this approach. Okay, I just want to mention that at Moffitt, there is a clinical trial of this, but they did not use gemcitabine. It was just the immunotherapy, but there is a second wave of clinical trial that will combine gemcitabine with immunotherapy. And acknowledgement, I want to thank my lab, my mentor, Dr. Kasia Rejnak, and Anna Anderson. She helped with developing the model, and other members of the lab and our collaborators. Thank you very all for listening. Thanks.
Of time, one very quick question back there, yeah.
That was very great, Kayode. My question is, how do you define the biology-constrained loss in the spin? And does that have to change with every histology you look at?
Does that have to change?
With every histology that you look at.
Okay, so we put them together. So let me go to the equation. So the biology loss here is actually, so we have these two time points. So we're just constraining the solution, those solution from pin at that exact time point to match that proportion. So it's just like an addition to the loss function. Yeah. And then there is some weight which pin is also trying to control so that none of the loss is dominating the other ones, because if you're trying to do this without correctly setting a weight to those losses, some are just going to dominate, and then you could result into overfitting and things like that. Yeah.
All right, thanks a lot.
Thank
One
you.
round of applause.